The measure of intelligence is the ability to change.
Hi all, Dr. Clark here again. This time we're going to talk about nucleic acids. So remember, we're talking about the four biomolecules, or the four main molecules that build systems in biology. We talked about proteins already. We're going to talk about nucleic acids. Then we're going to talk about carbohydrates, and then we're going to talk about lipids. So nucleic acids. For some of you, nucleic acid might be a new term. You've never actually heard or know what a nucleic acid is, but I'm pretty confident that most of you know what this molecule is. This is DNA. DNA is a type of nucleic acid. So nucleic acid includes DNA and RNA, and we'll cover, what the differences between DNA and RNA are. Now, over here, you probably have heard of these two individuals, James Watson and Francis Crick. They're the individuals that designed or discovered the structure of DNA, that DNA is double helix. Now, they're not the only contributors to this, but they're often the names that get contributed to discovering that DNA is double helix. They did other things like describe how DNA can replicate itself and other pieces of nucleic acid. We'll talk about the other players in DNA in a little bit as we progress. But, Francis Crick and James Watson. James Watson was an American scientist. Young guy, studied cells and kind of molecular biology before it was established as molecular biology, clearly because we didn't even know what the structure of DNA was during this time, and molecular biology deals with a lot of DNA work. And Francis Crick, he was studying hemoglobin and some other molecules, and eventually studied amino acids and a few other systems, but a European scientist, and these two got together along with the help of Rosalind Franklin and Maurice Wilkins and a bunch of other scientists that added to the material or helped along the way. They discovered the structure of DNA. All right, so let's dive in and look at nucleic acids. Like I said before, nucleic acids are, DNA and RNA, but before we get to that, what builds them? So remember, when we were talking about proteins, we said the monomer to a protein is an amino acid. Well, the monomer to a nucleic acid is a nucleotide. So the nucleic acid is the long polymer. These molecules, instead of building blocks for muscle and transport into and out of the cells, nucleic acids are information carrying molecules. Now, the information that they're carrying, which we call DNA or your genes, that information, the purpose of that information is to translate it into proteins. Okay, so your genes, the purpose of your genes is it's a code, it's a script, it's a manual to the proteins that your body can make and does make. So when we're looking at nucleic acids, it's not really a food resource, although you can break nucleic acids down, and we do. And you from that nucleic acid breaking down, you can get nucleotides, and you can reutilize nucleotides. You can get some sugar and phosphates and other nitrogen base materials that you can use in different biochemical pathways in different parts of your diet. But nucleic acids, when we're generally talking about them, we're talking about these molecules, DNA and RNA, and that allow for protein synthesis. So they're the actual written information that's telling your body what protein to make. All right, so let's look at the nucleotide. Let's look at the monomer and what does it consist of. Well, each nucleotide has a 5-carbon sugar. And now, depending on what type of nucleic acid it is, will depend on what type of 5-carbon sugar it is. It's either going to be deoxyribose or it's going to be ribose. And we'll look at this in a second. They have a phosphate group that is bound to that sugar. And then off the sugar, they have a nitrogenous base. Now that nitrogenous base is one of 4 bases in DNA, and then there's a separate base that's unique to RNA. And we'll cover these in just a second. Okay. It's the nitrogenous bases that give the nucleotide its unique type. Okay, so what nucleotide we have is based on the base. The phosphate group and the sugar, they don't change. If we're talking about DNA, there's no difference in the 5-carbon sugar. If we're talking about RNA, okay, the 5-carbon sugar differs from DNA, but there's no difference between the nucleotides of RNA in regards to the sugar or the phosphate group. So it's that nitrogenous base that changes. There are 5 different types of nucleotides, again, based on the 5 different nitrogenous bases. And like I said before, the purpose of nucleic acid is to code for proteins. Right? They're information-carrying molecules. So the order of that information gives you a different code and it gives you a different protein. We'll come back to this and I'll describe how that process works later. So let's look at the nucleotide structure. Again, a nitrogenous base, this is what's going to change, the nitrogenous base. Okay? So in the case of DNA, you have 4 nitrogenous bases. You have adenine, guanine, cytosine, and thymine. Okay. So that's where the nitrogenous base is going to be. It's going to be one of those 4 molecules. If we're talking about nucleotides in RNA, it's going to be adenine, guanine, cytosine, and uracil. Okay? You can see here that thymine is only in DNA and uracil is only in RNA. You can see that the molecules are fairly similar to each other in chemical form or chemical structure, okay, but they differ a little bit. And uracil is found in RNA and thymine is found in DNA. Now, if you look here, this sugar, this 5-carbon sugar, again, it doesn't change if we're talking about DNA. All DNA 5-carbon sugars are the same. If we're talking about RNA, then that 5-carbon sugar is going to be a little different. Now, what that difference is off the 2' or the second carbon in the ring. For RNA, there's going to be a hydroxide or an OH. For DNA, there's going to be a hydrogen. Okay, that's the difference. That's what makes DNA deoxyribose and RNA ribose. Then again, if you're coming off that 5' or that 5th carbon, okay, there's a phosphate group, and this doesn't change between the different types of nucleic acids. All right? So again, 2 types of nucleic acids. deoxyribonucleic acid and ribonucleic acid. They're very similar to each other, right? But there are a few differences that are important to kind of cover. Again, RNA uses uracil instead of thymine. Okay, we covered that. RNA is one strand. It's single-stranded. Now, that does not mean that when it copies itself or when it's in a different type of molecule, okay, like transfer RNA, that it doesn't wrap around itself. Okay? But again, it's still a single strand. Just because it can wrap around itself and form coils or helixes doesn't mean that it's not single-stranded. But if you're looking at a segment of RNA and you're looking at something and it's double helix in that region, it doesn't mean that it's not RNA. RNA can go double helix. It can fold on itself and form a double helix. But the big kicker that's going to be the difference is RNA is going to have uracil and it's going to have a ribose sugar. So again, yes, it's single-stranded, but that does not mean that it can't wrap on itself and look double-stranded. It can look double-stranded. Okay? Just concentrate on uracil instead of thymine and ribose instead of deoxyribose. All right, so let's talk a little bit more about the structure of DNA. We know the structure of DNA is double helix. We know it's double helix, well, because there was a lot of experiments done, and individuals like Watson, James Watson, Francis Crick, Rosalind Franklin, Maurice Wilkins, they discovered the structure as being double helix. But how it becomes double helix, or kind of the why it is double helix, is based on this individual's discovery or rule. Okay, this is Erwin Chargaff. Okay, Chargaff's rule is that adenine will always pair with thymine and cytosine will always pair with guanine. Okay. So again, adenine always pairs with thymine and cytosine always pairs with guanine. Okay. Now that pair, what holds them together are bonds, hydrogen bonds. Okay. Adenine is bonded to thymine with 2 hydrogen bonds. Cytosine is bonded with guanine with 3 hydrogen bonds. Okay. And this is Chargaff's rule. But because of this structure of A always binding with T and C always with G, okay, the DNA itself is a uniform thickness. It is three carbon rings thick everywhere because of the pairing that's going on. Properly aligned hydrogen bonds hold those bases together. Like I said before, 2 hydrogen bonds for adenine thymine and 3 hydrogen bonds for cytosine guanine. Now, often when we're looking at a DNA molecule, we're maybe considering the molecule looking like a ladder that's twisted into helical form. So if you think of a DNA molecule as a ladder twisted in a helical form, the rungs of the ladder, or the steps of the ladder, are these nitrogenous base pairs, these nucleotides paired up. The nitrogen base pairs make up the rungs of the ladder, the steps of the ladder. Okay. So you're stepping on an AT bonded by 2 hydrogen bonds if you're thinking of climbing a DNA ladder. Now, the actual stable part, more stable part of the DNA, which would be the edges of the ladder, or the actual arms of the ladder, they're built of sugar phosphates. Okay? So we often call it a sugar phosphate backbone. Okay? So that the ladder has this backbone or support system on the outside of the rungs that's made of sugar phosphates. Okay? They're bound together. That phosphate to sugar off the 5-carbon is bound with what we call a phosphodiester bond. Okay? Most people just call it an ester bond, okay? And they drop the phosphodiester part, or di part, and they just call it an ester bond, okay? But that gives that molecule a lot more support and rigidity to the DNA molecule. This will come into play when we start talking about how DNA replicates itself, because it's not the sugar phosphate backbone that's breaking up. Okay? It is this bond, these hydrogen bonds between nitrogenous bases that are being cleaved or broken in order for the molecule to be copied. Okay, so double helix. I mentioned, Watson and Crick discovering the double helix, but I can't really give them all that much credit because without the individuals pictured here, there's no way that they would have figured it out. Okay? So this is Maurice Wilkins. Okay? Him along with this individual who is Rosalind Franklin. These two individuals were the first individuals to picture DNA. Okay, so they got pictures of DNA using X-ray crystallography. And Rosalind Franklin is the first person to suggest that DNA, in writing, that DNA was double helix. Now, the reason why she's not given 100% of the credit for discovering the double helix form of DNA, okay, or the structure of DNA, is because Rosalind Franklin was very meticulous when it came to research. So even though she was fairly confident that it was a double helix, she needed to do more research. She felt she needed to do more research to figure out and be exact when it came to what's the shape of DNA. Now this guy over here we've already talked about. We talked about this guy when we were talking about proteins and proteins being in a helix form. Okay? This guy's name is Linus Pauling. Linus Pauling also had a big role in figuring out that DNA was double helix, because it wasn't Linus Pauling what Linus Pauling necessarily did. Now, yes, he described helical forms in proteins before we knew that DNA was a helix. So a lot of the research going into the formation of DNA, a lot of the techniques that were used to figure out the structure of DNA, they came from Linus Pauling. Okay? But what Linus Pauling did that really allowed Watson and Crick and Maurice Wilkins and Rosalind Franklin to decide kind of on this double helix is Linus Pauling said that DNA was triple helix. Linus Pauling wrote a paper that suggested that DNA was a helical form, but that it was a triple helix. Well, by doing that, it really spurred a lot of research in Francis Crick and James Watson and Maurice Wilkins and Rosalind Franklin, it spurred them to start looking at, well, is it possible that DNA is a triple helix? And indeed, it wasn't possible. So Linus Pauling, by publishing that, it was, a potential triple helix, it allowed them to decipher, no, it's not triple helix, but it's double helix. And so then they published that it was double helix, and they won the Nobel Prize. Well, Francis Crick, James Watson, and Maurice Wilkins won the Nobel Prize for the discovery of DNA. Rosalind Franklin did not win the Nobel Prize, and it's not because her work and because she was a woman. Part of that was an issue at this time, but Rosalind Franklin had passed away. She died before they received the Nobel Prize, and you can't win the Nobel Prize if you're dead. So that's why you don't see Rosalind Franklin's name as the Nobel Prize winner for the discovery of the structure of DNA. All right, so we know now from other research, some of this was done by Francis Crick, that structure of being double helix really helps with the functionality of copying itself, with the functionality of being able to copy small segments for protein synthesis, or being able to copy your entire genome for cellular reproduction. Okay? And it is these hydrogen bonds, we talked about hydrogen bonds before, these weak hydrogen bonds, they're easily broken. Now, DNA, there's 3 billion base pairs in our genome, so there's, roughly 6 billion, far more than 6 billion, but at the minimum 6 billion hydrogen bonds in our genome. That will make that structure very strong. But individually, hydrogen bonds can be broken easily. In fact, there's a single enzyme that we use to break those hydrogen bonds. It's called helicase. And it comes in and it breaks the hydrogen bonds between A and T, C and G, and it unzips the DNA so it can be copied. The other nice thing or unique thing about DNA is DNA runs anti-parallel to itself. So one strand runs in the direction that we call 5' to 3'. So from the 5th carbon to the 3rd carbon. Okay? The other strand runs the opposite direction. It runs from 5' to 3' in the opposite direction. So this means that DNA is a mirror image of itself. Okay? The strands are flipped. Okay? But because of that, it makes for copying your DNA much easier. Okay? And so DNA contains 2 copies of all the information. If your molecule has 2 copies, one on each strand, it means the information can be more accurately copied and passed on to the next generation with fewer mistakes. We'll talk about these mistakes as we progress. These mistakes are often called mutations. Okay? And mutations can be in quite a few different forms. You can have chromosomal mutations or gene mutations or nucleotide mutations. And we'll come back and we'll talk a little bit about this because it's important for modern medicine and modern technology to know how these mutations might occur and what's the benefits and costs to these mutations. Okay, with that, we're going to kind of wrap up that nucleic acid. We'll come back. Okay, we're not done with nucleic acids. We'll come back and talk about DNA. We'll come back and talk about genetic therapy. We'll talk about CRISPR, Cas9 systems, or genetically modifying an organism. We'll come back and we'll talk about a little bit about pedigrees and how you inherit certain traits, or what we call Mendelian genetics and non-Mendelian genetics. Okay, so we're going to reapproach a lot of these DNA topics because DNA is big right now in society. It's huge. If you can manipulate certain plants to have them grow certain characteristics that you want, if you can manipulate certain animals to have them produce higher nutrient loads in whatever it is, meat, milk, whatever it might be, eggs, we're going to explore what's out there. And this is all based on genetic or DNA work. Okay, next time we're going to talk about carbohydrates and lipids.